Okay, hello everyone, I hope you can hear me. Thanks again for joining us to hear about the role of proteomics in understanding COVID-19. Today we're joined by a return speaker, Dr. Christian Muench, to discuss his further work in this area. But first of all, we have a few housekeeping things to go over. So as always, we're using our Slack channel for questions and discussion, so please join us there to ask the questions and share any thoughts and discuss the work. The Slack really helps us to prioritize the questions that others want to hear the answers to, so use the thumbs up to let us know the questions that you would like to hear answered. Slack will also allow the speaker later to answer any other questions or follow up after the talks have finished. And for those needing a certificate of attendance for this webinar, details will be available on how to get this after the last slide. And once again, we'd like to say a big thank you to the European Proteomics Association, the British Society for Proteome Research, Young Investigators Proteomics Club, and the London Proteomics Discussion Group Committee for their help and support in setting up this webinar. Also thanks to the London Biological Mass Spectrometry Discussion Group, the London Metabolomics Network, and the News in Proteomics Research blog for promoting this event. We're also really grateful for Imperial College London for providing us with webinar support. And a huge thank you to Christian for agreeing to give us his time today to give us another talk. We're also pleased to announce that our next webinar will be on Friday, the 10th of July at 2 o'clock British Summer Time, featuring Professor Leonard Martins, and he's going to be talking about proteomics and the role of machine learning, how the data rich get richer, machine learning as the key driver of MS informatics. And our next COVID talks will be on the 24th of July from Professor Andreas Pickelmeyer and Dr. Jerome Demers. So, Christian Muench studied biochemistry at the University of Tübingen and the Max Planck Institutes in Martinsried and Tübingen. He obtained his PhD from the University of Cambridge in the UK, and his graduate work earned him the prestigious British Neuroscience Association Postgraduate Award. After his graduation, Christian joined the laboratory of Wade Harper at Harvard Medical School with an EMBO long-term fellowship to study protein quality control in mitochondria, and particularly the mitochondrial unfolded protein response. Christian started as an independent group leader at the Institute of Biochemistry in Frankfurt in December 2016, and his main research interests focus on mitochondrial quality control, in particular determining the process involved in the mitochondrial unfolded protein response and its role in human diseases. And today we're going to hear from him about his latest work employing a SARS-CoV-2 infection system in permissible human cells to study signaling changes by phosphoproteomics. So it's over to you, Christian.
Yeah, thank you. Thank you for having me here today. Again, a nice repeat opportunity, but also thank you all for everyone who logged in and is listening to this talk and is taking some of your time to listen to the things that we've been doing in the recent past. So as mentioned, I gave a talk a couple of weeks ago, and some parts of that I will have to repeat again, but especially as some of you are probably joining for a second time now, I'll focus much more on things I haven't been talking about last time. And I think there should be some nice new flavors in there. So just as a general point where I'm coming from is where the interest of my lab is, as was mentioned, is to gain an understanding of stress responses in cell. And they're mainly this idea that stresses can occur in different places in the cell, from the outside, in different compartments, cytosol, protein aggregation, and there's a whole host of cellular responses that are activated upon those stresses. And I think it's really important to gain kind of cell-wide views of what's going on. And clearly proteomics is one of the best suited tools to be able to do that, to really gain this kind of on a molecular systems biology level understanding of what's going on. And what we're focusing on a lot is, of course, there's often signaling responses upon stress that leads to changes in transcription and translation and various outcomes from cell cycle changes to metabolism and so on. I will introduce quickly some of the technologies that we are using in the lab to do proteomics. I will not introduce proteomics itself. We do bottom-up proteomics, which means whenever we have samples of interest, you extract the proteins, you digest them, and it's really peptides that we analyze. And a tool we like a lot is these so-called tandem mass tags, which are essentially like barcodes that you can use. Currently, there are up to 16 of those, where for 16 independent samples, you can create your peptides. And then you can label every single peptide in your solution with these barcodes and 16 different flavors, so you can distinguish later on from which of the 16 samples the peptides came from if you pool them together to analyze them. Yeah? So the whole strength here is really you can pool the 16 samples together to one tube. It saves a lot of machine time and allow much more easily to do fractionation approaches so that you get a deeper proteome overview without creating artifacts just from doing fractionation so many times. Here it happens with the same mixed sample, so it's not a big problem. Then you can analyze that in the machine. And here this overview of these TMT tags, the NHS esters, so how they work is they modify the N-termini of peptides and the lysines on the Z-terminal end, if there is a lysine there. And in the machine, these molecules, which all have the same mass, can get fragmented across this dotted line, and we can then measure these reporters that have different masses and tell how much of the intensity in your sample came from every individual sample that went in there to allow really quite accurate relative quantification. The great part is that these molecules do all have the same mass, they're isobaric. So at first, if you analyze them in the machine in your first MS1 scan, where you just get the mass of your peptides, there's still only one peak. You know? So it adds up, it sums up. And the other great side effect of that is that now this one signal is composed of the 16 samples, which means you also need less sample input to get to these intensities that you require to be able to measure them in the machine later on. So we use this a lot. There's different flavors to it. That's something that was found 10 years ago already, extensively studied by the Gigi lab, where it turned out that there has always been some problem if you do wholesale proteomics using TMT, stemming from co-isolation interference. The idea is that there, typically you do your MS1 scan, you select the peak, and then you would do analyze the fragments in the MS2 and also the TMT, but because of course you cannot get your window precise enough just around the peak you want, you will always co-isolate other things, and that can lead to compression of your ratios. And this is kind of shown here. In normal conditions, if you have mixed ratios that you know what to expect of your 10:1, 4:1, and 2.5:1. In red, this is without interference. What you would measure, however, with an MS2 approach where you have this interference, you can see that the ratios are quite compressed. And the way to overcome that was really to introduce an MS3 method, where in the MS2 first you do CID fragmentation to just get the peptide sequence, and then in an additional step do HCD fragmentation to fragment the TMT reporters and quantify them here. Yeah, and with that you came back to quantifying pretty accurate levels here. And I introduced that because we'll get back to that later in the talk and some of the things we've been doing. I introduced that last time, and it's really something where we have been stuck for a long time. I mentioned before, we want to see cell-wide changes to stress. And particularly when it comes to proteins, one of the issues is, of course, if you look at total protein levels, it typically takes quite a while for them to change. So in some ways, it's not a good way to really very acutely measure changes upon a stress. It's quite different, though, if you look at translation. Yeah? And in translation, you can really monitor the situation, the current moment. The problem, though, is it's difficult to look at translation. And one of the difficulties is shown here. And this is not proteomics data, but this leads to ribosome profiling. And here's an example for mTORC1 inhibition. Yeah? So mTORC is really one of these central cellular hubs that integrates a lot of different stresses and has quite severe outcome and output towards the cells, mainly that it leads to inhibition of cellular translation. So that's shown here. If you inhibit mTORC1 with taurin1, see that 60 to 70% of cellular translation are gone. The same if you look at polysomes, you can see that most of them are gone, so ribosomes here are not translating anymore. And as I said, standard method for a long time, and it's still, I think, the most important tool that's out there is ribosome profiling. However, if you look at the ribosome profiling data set from this kind of example, and this is taken from this paper here, you can see that this is normally distributed around 0. So what that essentially means that on a global scale here, it would look as if globally there's no shift in translation. And it's very clear where this is coming from. There's no mistake or error being done. It's just a normalization bias that happens in these kind of experiments where you would purify polysomes from here and from there. you would normalize the amount of input to PCR. So in the end, it looked as if they're both at the same amount in there, and you get this distribution around 0. So it's still useful to find these outliers that are changing, but not so good to get a global view under these really severe conditions where effects are strong. Yeah? So under other conditions where translation globally is not affected much and there's only specific targets, this will not be a problem. But we thought there might be ways with proteomics to be able now to specifically see these kind of issues. And we turn to something that, of course, has been there for many years, and that's pulse stylic labeling. So the idea there is you have cells growing normally in the typical naturally occurring amino acids, so light peptides in a way, and then at a time point 0, you shift and changing your lysines and arginines to heavy isotope versions of those. So over time, newly translated proteins will incorporate these heavy amino acids, and you can monitor that over time for different proteins and look at those kinetics. In principle, that works pretty well. It doesn't work so well, however, for what I mentioned before, if you want to look at specific stress responses, because the problem there is you don't want to look after a day. You really want to see an hour, 2 hours after you're stressed to see what's happening. And where we are in that case is inside this red box where the amount of heavy peptide compared to the remaining protein that's still there, so the light protein, is really, really low, and it's very hard to measure that. And so that leads to a situation like this, and this is actually even underemphasizing the problem that you have a heavy peak at a very low ratio compared to the light peak. This is often really just 1% and in that range. And in our typical approaches using top 10, top 20 methods, something like that, and DDA, we would usually not select this peak for analysis. So there is kind of an not absolute measurement threshold, but it's something in the fact that you usually don't pass, and it's even on a software end a problem to identify these isotope pairs if the ratios is quite far from each other. So in the end, these heavy peptides will not get selected, they will not get quantified, and you just don't get information about the translation. And how we thought it might be able to solve that is combining this dynamic SILEC with TMT in a method that we call mePROD, for multiplexed enhanced protein dynamics proteomics, where we use the TMT and set all the samples add up, and for any given peptide, which allows us now to include one boost channel that is 100% heavy labeled. It boosts the signal, it changes the ratio between light and heavy just to a point where now you're above the measurement threshold, and it allows you to select this peak as well. And then, of course, on the TMT reporters, you still get a very good relative quantification out of that to get an information about what your translation changes was. And this is what it looks like. Here's the boost channel. We can do our quantification relative to that. And we also have a baseline channel in there to determine the noise and overcome ratio compression in the end. And this is the work spearheaded and done by a great PhD student in the lab, Kevin Glann, with the help of Georg Tascher. And he showed here in HeLa cells, so that also works in the cellular system where we can label for 15 minutes, 30, 60, 120 minutes. And you can see that we get a very robust quantification, is linear over time, replicates are tight, so it works quite well. And we applied that, and here's just an example, and I will end with that then. This is the Torin one again that I mentioned earlier. Now it's our data. We can globally see this reduction in translation. We could determine which proteins are reduced upon these stress conditions. And in the paper, we try to compare how different cellular hubs control protein translation here, specifically whether the integrated stress response and mTOR overlap, and they do quite intensively. And the nice part is, because it's largely standard mass spec methods, we can really scale this up, do a lot of samples, and look at a lot of conditions. So with that, I will end it there, but I think it is important because this is a method we use later for our SARS-CoV-2 research. So here's a summary again from this dynamic SILEC approaches and the mePROD that overcomes this issue of not being able to determine these heavy peaks. What it ends up with, we can measure highly acute translatomes, so the nascent chains. There is no normalization bias. We need very few cells, so we do ribosome profiling in the cell in the lab. It still remains a very powerful tool, but you need a lot of cells. For us, typically about 20 million, and it's quite expensive. So here we can work with less than 100,000 cells. It costs us about 1/10 of ribosome profiling. And as just mentioned before, you get a very high throughput, especially compared to ribosome profiling, and a full proteome coverage. Yeah? So quite a couple of thousand proteins. And yeah, that's also what I introduced to you last time. And then we also came up with this other thing where we realized, and I mean, this is still an ongoing problem with all bottom-up proteomics, that we waste a lot of machine time on measuring peaks that we don't care about. And this is exemplified here. If you do a typical data-dependent acquisition, here it's supposed to be these pairs of light and heavy. And of course, if you do a top 10, top 20 method, it will just select the most intense peaks. So essentially it could be all of the ones here, no matter whether they're the gray ones that don't have an isotope pair or not. You will waste a lot of machine time, and there's not enough time to go around for the pairs that you really care about. And then something that was quite interesting is that we saw in the new or newer machines that came from Thermo Fisher, in our case it's Lumos, there's machine logic algorithms built in that can target a mass difference and can determine on the MS1 surface scan already whether there's isotope pairs here, and can then only select those for an MS2 analysis. Yeah? So we would drop all these gray peaks that we don't really care about, and we can focus specifically on the pairs that we want to make sure that we really have them all the time. Yeah, and this is especially important because here you can see again the heavy are very low ratio compared to the light. And we need to make sure to have them because otherwise we cannot get any information at all about the translation rate. So an experiment to show how that works and to validate whether this is possible is again this kind of mePROD setup where we used artificial heavy to total ratios of 1%, 5%, 10% in triplicates with the boost channel and the baseline channel, and then we used kind of standard data-dependent acquisition, both MS2 methods and SPS MS3 methods, and then the same with this targeted mass differences I introduced here. And it was shot on a Lumos and then analyzed in Proteome Discoverer. And what we found was quite great, because if you compare both on MS2 here and on MS3, we got about a 2.5-fold to 3-fold increase in identified heavy peptides. So this is really, for us, a big step forward. It means you have to shoot much fewer fractions and you can get much deeper in your proteome. And we did further validations. Here's just an idea of getting lysine containing peptides, so the ones that have heavy or light lysine in there. You can see here with TMD now that most of our peptides, or a large fraction, now also contains the heavy lysines, while before with the DDA approaches without TMD was just a small fraction. Generally the MSMS scan numbers don't change. However, the mean MS2 summed intensities go down with the TMD approach. I think that's not surprising, because what the TMD does, it allows us to select all these low abundance peaks. So you would have much more of those compared to your top 10 method. So of course, also the summed intensities will go down a bit with that. And we then looked generally how good the quantification is that you can get with that. Here's the comparisons, again, this 1 to 5 to 10 ratios across all of them. It's written down here, and comparing MS2 and MS3 methods with DDA and TMD. What we saw there is that there was actually, the ratios were quite high for these TMD methods. And we thought it probably is not something experimental. There's got to be a technical explanation for that. And we thought about that for quite a while. And of course, one of the issues that you can have, depending on how you set it up with your TMT experiments, is that the reporters are not necessarily direct representation on how much you had in your MS1 peak. Yeah? So here we don't have, like in label-free experiments, a quantification of the MS1. Instead, you tell it to target how many ions you want to get and how much time you can give it. So the ion intensity you get for TMT doesn't necessarily directly reflect how much input there was originally, because there's also maximum injection time. So we try to compensate for that by adding an extra step in between where we normalized for ion injection time. So that gives us an idea how many ions were there in the first place to overcome this distortion we otherwise get. And this is what it looks like now. And you can see in DDA, it didn't really change much, but with TMD, we're getting much closer to the ratios. And actually for TMD MS3 measurement, we get very close to the perfect expected ratios. But mind you, again, this is if you want to get an understanding in more absolute terms to get this 10 to 5 to 1. However, if you want to just relatively quantify, which is typically in most cases what we do, this all doesn't matter much. Both MS2 and MS3 methods with DDA or with TMD, they all can quantify this fold changes between two samples very well. Yeah? So it's just if you want to get this 10 to 5 to 1, you have to put in more efforts. If it's just seeing this 5 to 1 ratios, it's much simpler. One interesting side effect that we had with all this, and we discussed it a little bit also in the last presentation, is the issue of co-isolation and how well to determine it. And what we realized with this approach, one very nice side effect is that now we had a method, and there hadn't been one so far, where we could experimentally measure the co-isolation interference. The idea would there be, if you isolate the peak of interest here in MS1, we can have co-isolates that come along with that. But because we have this this noise channel, we know that there cannot be heavy peaks in there. We know this is absolutely empty. So from there, we can determine how many co-isolates came across that gave us a signal in there. We can measure that. That's shown here with a measured isolation interference. And I don't think we have to take this super far, but for us, quite an important point to make is that there are approaches, for example, in Proteome Discoverer, to determine co-isolation interference based on the MS1 scan. And you can see here, this doesn't correlate very well. So I think all to say from this is that I think all of these predictions of co-isolation interference they may use in TMT experiments to overcome these issues. Still take them with a grain of salt. I'm not sure how accurate they are, and it's probably not ideal. The other point, though, to say is, of course, in our experiments, these can suffer much more from co-isolation interference in the whole cell proteome, just because we focus on these very low intensity peaks. Co-isolation that comes at a low level across all peaks, of course, is higher relative to the low peaks than it is in the light peaks. So we just have more co-isolation interference here. And that's also what we see if we actually measure now in our setup, this isolation interferences with the TMD, where we have many more of these low peaks. We also see more of an isolation interference there. Yeah, we try to overcome some of these co-isolation interference issues by making the isolation windows smaller. And we tested 4 from 0.4 to 0.7. And you can see that the heavy peptide identifications don't change much, but the interference goes down significantly, and also the general coefficient of variance is reduced a lot by making this window smaller. So that's where it is. We had this mePROD method that I explained to you before, where you use this pulse Silac labeling combined with TMT, and then the boost channel to specifically boost the heavy peptides. Now we combine that with this machine algorithms in the Lumos to only detect the peaks we care about, to get at about 3-fold increase in identification rates. Yes, that's about that for it. If you want to find out more about this study, you can read up down here in this paper. So with that, we get back to our stress responses and also to COVID-19. I mentioned stress can occur in different places, including viral infection and bacterial infection, which is all actually processes that we are currently studying in the lab. And our work started with SARS-CoV-2 suddenly arriving in Frankfurt, where I'm located, on February 1st, because there was an evacuation flight from China that brought about 150 people back from Wuhan in China to Frankfurt, which is, of course, the biggest airport in Germany. So typically the place where anything special happens when it comes for importing things to airplanes. So 2 of these patients, of these people actually were infected with SARS-CoV-2, and the virology department at our university managed within days to isolate these viruses and get a cellular infection system going. So this has really been a fantastic collaboration with the Institute of Virology in Frankfurt, and is spearheaded by Jindřich Cinatel and also Sandra Zizek, and run a PhD student, Denisa Vojkova, and Benjamin Koch, who is a medical doctor. And what they managed very quickly is to set up this cell infection system. So this here is using CACO2 cells. So these are colon cancer cells in a mock infected conditions, they look like this. If you infected them with SARS-CoV-2 for 24 hours, you can see that they're very clearly showing a cytopathic effect. So the cells are dying, they're opening up, they're releasing virus, and we have here a really working infection model. And to confirm that this worked, we used these CACA2 cells, infected them with SARS-CoV-2, and then took out some supernatant over time and validated the release of viral RNA into the supernatant. And you can see that here. So I think it's quite clear from there that we had a robust model of infecting cells and that we could then study with our tools. And here too, if you want more information about that, you can read up in this paper. Also want to say that we made all the raw files available. We have a tool on our website where you can look for gene, protein, name of interest, added all the Excel tables to the paper that really everyone has a chance to go through that data and use it in research and try and help solving the current crisis. So for us, how we set up our experiments, we used those CACA2 cells, infected them with patient isolates, and then used this mePROD method where we pulse labeled with lysine arginine, and after 2, 6, 10, and 24 hours, we harvested proteins, labeled them with TMT, shot them in our mass spectrometers, and look for proteome changes and translatome changes. I will go through it quickly and just give you some examples because it was the topic of my last presentation here, but I think it's still good as a refresher, and especially for everyone who's here who didn't hear the last talk to also be familiar with that. Just general overview that it works. Here's the translation kinetics of viral proteins, and you can see that for these 5 viral proteins that we detected, the translation increases over time. And as labeling was always done for 2 hours, this consistent increase in rates actually means it's not the accumulation of the protein, but indeed the translation rate increases over time. Yeah? So there's more produced at this time point than at this one. And then came one of the really first big surprises for us, where we globally looked at what happened in translation after SARS-CoV-2 infection. Because for a lot of viruses, it's assumed there's actually not that much data out there on clear infection systems with kind of a native virus on a cell to look globally at translation. But most of them suggest that there's often a very severe reduction in global translation rates. And that has also been suggested for SARS. However, when we looked here, we didn't really see much of a difference. Yeah? So after 10 hours, that was the maximum. We saw about a 23% decrease in translation. 23% is still a lot, but it's far away from the 90%. And I think this is alone is something that will be really important to study in the future, get a better understanding what actually happens after viral infection for different viruses, for different cells, and to use these kind of methods and not overexpressing viral effector proteins or just capsids or so on. And what we found in the data, and I condensed some parts here first on the translation side, kind of probably explain to some extent why translation isn't shut down and not reducing for the host proteins, even though all these viral proteins need to be produced now, is that there's quite a high increase in all kinds of terms related to translation, with ribosome translation, elongation, initiation, and so on. So it looks like the host cell somehow drives up its own translation capacity after infection. And we thought, okay, if these pathways are so clearly important and increased upon infection, can we use inhibitors of translation like cyclohexamide or amidine to block viral replication in cells? And indeed, that was possible. So here is shown inhibition of viral replication. And in both conditions, we saw this inhibition at below micromolar IC50s and at non-toxic concentrations in those cells. And then we looked further. I mentioned this one particular example because it comes back in the phosphor data, where we now looked at the whole cell proteome changes, especially in cluster 2, things that were going up. Yeah? So these 3 here were the 3 triplicates, results after 24 hours. We just looked what increased. There were a whole range of pathways that we tested, but quite strikingly, one of them was carbon metabolism, also focusing around glycolysis. And I thought we might check 2-deoxyglucose, which is a well-known and studied inhibitor of hexokinase of glycolysis in the end, of the use of glucose. And we saw prevention of viral replication in cells when treating cells with 2-deoxyglucose. And that was quite striking to us because, I mean, it might seem harsh to block glucose, but nevertheless, it's treatments that are being used. has been shown even in animals. And in rhinovirus, you can use 2-deoxyglucose to impair viral replication. And 2-deoxyglucose and pre-drugs are actually tested in a whole bunch of different animal models for cancer therapy. So I think there is potentially a way forward, and you can really use very high concentrations of that stuff in animals, largely because it's metabolized quite quickly. And one other interesting point we looked, this is again on total protein levels, at the profile of what changes. Here is shown for the 9 viral proteins we saw. Again, they increase over time, so here there is a real accumulation, and we followed them in the host cell, when host cell proteins behave in a similar way. And what we found in these types of cluster analysis was mainly process involving nucleic acid metabolism. Probably not surprising, of course, the virus needs to reproduce its genome, so it also requires bases. Even though, I mean, we saw when we compare different inhibitors that there is some specificity, but what we did find is that there's this one inhibitor, ribavirin, which inhibits IMP dehydrogenase, and at low micromolar concentrations, it had inhibited replication of the virus in the cells. And for us, what was really great here at this point is that some time after we put this online, there was this also now efforts from clinical sites where there is now about a month ago, so the first clinical studies initiated using ribavirin. Here it's with a trade name Virasol, which is an aerosolized ribavirin that you can spray into the lung, and it's being tested in COVID-19 patients in Canada. So I think as a basic researcher, that's kind of nice. You have here some basic data in the end that led us towards the translational aspects, identified drugs based on our proteomics data that could then be used and transferred now into the clinics. So in the summary of that part, we had this cellular infection model for SARS-CoV-2. We studied whole cell proteome differences and translation proteomics differences. We found 5 pathways mainly that were really important, upregulated. I didn't mention all of them. It was translation, nucleic acid biosynthesis, but also splicing, proteostasis, and glycolysis. And we also saw that the global host cell translation remains largely unchanged. But what we found is that for every single one of these 5 pathways, if we inhibit them, so these pathways were increased upon infection. If we inhibited them with drugs, we prevented SARS-CoV-2 replication in cells. So that was the end point of that study. And there was still, for us, one big thing missing, and that's kind of these gray arrows underneath here. So we now know that these pathways are changing, but we don't know what are the signaling events that may lead to these effects. So we really wanted to find out how does signaling change in cells upon infection. And in some ways it can be a bit more complicated. You need more material to do this kind of stuff, because for us proteomics people, of course, the way to address this is phosphoproteomics, which requires enrichment. And up here is the experimental setup that we used for these experiments, still using this very robust CACO2 cell system. And should also mention at some point that you need permissive cells to be able to infect them with the virus. And that depends very much whether the correct surface receptors are available for the virus to be able to dock on and enter into the cells. And very often, and I think this is still something ongoing, it's hard to find really good models and establish them in the right way. So I think largely what we and others are doing is using cancer cell models, often overexpressing proteins like ACE2 to make them even permissible. But I think there's a lot of research coming from there that will ultimately, of course, like to later on validate once that becomes available in up to even animal models. But what we did here is using our cell infection models, mock infected or SARS-CoV-2 infected. We did 5 independent biological replicates for both conditions just to get a very strong statistical power in there. We pooled them all together just because we didn't have that much. Created one 10-plex TMT from which we took some out for total proteome and did a phosphopeptide enrichment with the rest. And even with this big experiment, 10 different samples, what we ended up with here is about 10 micrograms of phosphoeptide. So it was quite limiting, but it was enough to still fractionate that into 4 fractions to shoot with an LC-MS2 method. The total proteome got much deeper with 24 fractions that we then analyzed later on. And what we found in there is the proteome changes that I mentioned before, but more strikingly now in this study is changes in the phosphoproteome. And we identified over 15,000 sites. And as you can see here, there's really a lot of regulation, a lot of peptides that were down or upregulated here in blue and red respectively. And this was mainly comprised of serine phosphorylated sites, which was kind of given by our methods because we used IMEC approaches. And then, of course, you don't get much tyrosine phosphorylation in there. And nevertheless, there was a lot of stuff there. Here's just a general validation looking at the total proteome and the phosphoproteome by a principal component analysis. And I think it's quite striking that there is a big difference between those two populations. It is driven on the component 1 by the infection, the replicates cluster together, and they're quite significantly different both for total proteome and for phosphoproteome. And then we looked more detail what's going on. First, we focused on the viral proteins, just seeing what gets phosphorylated there. We did identify a good number of phosphorylation sites in the different proteins. Unfortunately, for a lot of these proteins, their complete functions are often not clear, and especially not how they're being regulated and what specific phosphorylations actually for them mean. That's something we will have to look into in the future. But I think there's still interesting aspects, like in those two proteins here, protein 3A and membrane protein, you can see that the phosphorylation happens in the cytoplasmic domains. And what we found quite interesting is here for nuclear protein N, where there's a crystal structure available just of part of this RNA binding domain here, just this bit. And we found 3 phosphosites in there, and they all 3, if you look then in the structure, are clustered around the same area in the protein. So I think makes it quite likely that there may be a specific motif that leads to modification. So there could be a specific kinase required to do that. It also means that there might be a specific interaction surface that gets changed upon phosphorylation and could lead to specific functions afterwards. I think where it's easier to look into and get more specific information is now, of course, looking at the host cell proteins. There we understand much better what happens. And I mentioned one example of one pathway where we found a lot of phosphorylation differences, and that is in carbon metabolism. And I mentioned this because in the previous part of the talk, I told you that inhibition of glycolysis was sufficient to prevent replication. And it's quite striking that also generally upon infection, we see a massive increase in phosphorylation on many of these sites and a lot of different components of these pathways. So that might explain to some extent why on translatome base and protein, total proteome base, we saw this increase in carbon metabolism might to some extent be driven by these phosphorylation events. And we also did, of course, kind of more global analysis generally across the whole proteome. And there we condensed the information a bit on the protein and protein level changes, and condensed all the phosphosites in a protein to get kind of a single value that we could correlate with the protein change. And I think if you do correlation maps here, you can see that there's distinct clusters, especially here, the one that we call cluster 1, cluster 2, and 3. And it's striking if you look into them. So this is cluster 1. As you may see here from enriched, this mainly involves all kind of signaling pathways in the cells, involving GDPases, EGF receptor related things, endosomal uptake, and a lot of things that really make a lot of sense in terms of signaling events upon infection. And we also compare to what extent these changes are driven by changes in protein levels or phosphorylation. And you can see they're looking at ratios that is very clearly, for this cluster here, driven by the modification by phosphorylation. While if you look at another cluster, here's cluster 2, it looks quite different. We have here subpopulations that are driven by the modification and some more by a decrease in protein levels. And in this particular one, it was evolved around cell cycle changes, transcription, and so on. But what we were really after in this study is to, again, get to this translational aspect, to identify pathways where there's a significant change in signaling, yeah, so increase, decrease in phosphorylation of a whole pathway after infection. And we cared particularly about pathways that are manageable for treatment, so where there are FDA-approved drugs available. So we overlapped maps and cluster them together, FDA-approved libraries of compounds, of which we know the targets, together with the changes we saw in the signaling. And that was the outcome of that, very strongly focused around EGF and generally growth factor receptor signaling. Here, these blue circles are all the proteins that we saw changing, and here the yellow routes are different drugs that hit at different points in these signaling pathways. So with that, we were wondering, whether this is a general feature upon infection, and here it's just shown for EGF receptor signaling. It's this two main downstream signaling pathways, from RAS-RAF signaling and AKT signaling that goes into mTOR. And from the receptor to the downstream targets, we saw increased phosphorylation of those, consistent with activation of the signaling pathways. There were also other aspects like receptor endocytosis, which is related, that was increased. Also changes in cytoskeleton modeling. Of course, quite interestingly, also changes in the integrated stress response, which could be here a bit of a fight between the host cell and the virus towards trying to shut down translation to prevent the virus from replicating in the cells. But we then asked, if now we have this pathway, can we pharmacologically inhibit this pathway to reduce or prevent viral replication in cells? So it's similar assays as I showed before for this translatome project. We infected cells with SARS-CoV-2, and because we saw changes in different growth factor receptors, we thought it might make more sense to inhibit the downstream signaling, these two pathways that are similar for these different receptors. So we took these 5 different compounds that inhibited different stages here, and then assessed them independently, what they do to viral replication. As you see here from these curves, all 5 prevented viral replication in our cell culture system. The IC50s are given here. I also show the maximum achievable plasma concentration in people as far as there were available, that you get an idea how close these are to what we need. And it's actually for most of them very, very similar. And we also validated by different means. Here's looking at the release of viral RNA into the supernatant, and this was also prevented by these 5 drugs. And here we stained the cells for viral components here as double-stranded RNA, so the viral RNA. And may appreciate, while under non-treated conditions here, you have a massive amount of these double-stranded RNAs in the cells, and you can see cell death, a cytopathic effect occurring. With different concentrations of these compounds, this doesn't happen anymore, and the cells look really fine. There's no toxicity going on under these conditions. So what we had there is that it appears that upon normal conditions, where we didn't interfere with the system, that upon infection there was quite a modulation and activation of growth factor receptor signaling, activation of this PI3 kinase and MAP kinase pathways, and we get a high replication of the virus in the cells. However, if we inhibited one or the other of these pathways, we wrote low replication to no replication of this virus, we prevented these effects. And this data can be found. We put it on a preprint on bioRxiv. Again, even the raw data we made available already, feel free to go through it, look through it, whether you find something interesting, feel free to also let us know. So with that, just a summary. I will go through it quickly. Also, we're coming to an end time-wise. I showed you the development that we did to produce this new method we call mePROD, to very acutely being able to measure changes in the translatome, published here. Showed to you this new use of instrument logic to improve those measurements. And then we went into this COVID research. We first used translatome and whole cell proteomics to look at pathways that are modulated upon infection. And we found inhibitors for all these pathways that prevented viral replication in the cells. Data is here. And then we went to carry out phosphoproteomics in the cellular infection system, and we found most strikingly changes in growth factor receptor signaling, and inhibition of growth factor receptor signaling prevented SARS-CoV-2 replication in cells, and the data can be found here. So with that, there's a lot of things to give away. This work was really all, you may have seen from the names, spearheaded by this brilliant PhD student in the lab, Kevin Klan, with a lot of support from Georg Tascher, and also our quantitative proteomics unit in Frankfurt that I'm heading. This would all not have been possible without this fantastic collaboration with the virology here in Frankfurt, with Jindrich, Denisa, Benjamin Koch, and Sandra Zizek. And also would like to mention my mentors really helped me a lot with all kinds of advices, Ivan Dikic, my department. Great thanks goes to the different funding bodies for this research and generally for my lab. Here's information if you want to find out more. You can also access the data through here. And if you're on Twitter and you want to find out what we're continuing to do, join me and you will see. And with that, I'd like to thank you very much for joining this presentation, and I'm really looking forward to the questions. Thank you.
Great, thanks very much, Christian. That was excellent. There's quite a few questions come in on the Slack channel, so I think we've got some time to go through them. The first one is, what is the relationship between the rates of transcription and translation?
Yeah, I mean, this is more general, I guess, not so much in terms of the infection, but I think this was one of the really striking things in the very beginning when ribosome profiling was discovered, and that Jonathan Weissman worked a lot on, that they don't correlate very well. Let's put this as a bottom line, yeah? So of course, if you have highly abundant proteins, you also need a lot of mRNA for it. But I think it's two very useful pieces of information for different questions. In my mind, transcriptome is really important to understand the upstream signaling, activation of transcription factors, and so on. But proteins are the ones that in the end have the function and do the output. And that can be driven by many other things, changing in translation rates and degradation rates and so on. So I think it's really powerful to have both pieces of information. But if you want to have a functional understanding, I think, and this is a proteomics group, so I guess most would agree, proteomics is really important there.
Great. There's a couple of sort of more technical questions. How necessary do you think the MS2 is for the heavy or lower abundance peak in a dynamic SciLac experiment? And can the lack of MS2 on both peaks be overcome by a retention time correlation?
Yeah, it's a good question. We tried around a lot how to do that. It's also, I think, still general the question where they go from, excuse me, I just need to drink. They go for MS2 or MS3 approaches because the ratio compression wasn't so bad. I think MS2 is really useful. We still have very complex spectra, so I think in our hand has been very hard to work with retention times and things like that. And the issue that remains, I mean, in all the images I showed, the heavy peaks are often still much, much higher than they are in reality. And I think you really need to somehow boost them and very specifically look for them to get them, because otherwise you're more in the range of the top 40, 50 to get there. And looking for retention times, and it's also too complex, really, it doesn't get us there. And of course, with the SciLac, our spectra are twice as complex as a label-free spectrum would be. Yeah.
So can you or have you coupled your new paired isotope detection that you were discussing on the Lumos with the new features of the real-time search for the SPS MS3? For making sure that you actually only acquire MS3 on the good MS2 data?
Yeah, we would love to. We don't have the machine for it. That's the current state. We have a Lumos, which doesn't do the real-time search, and I would love to have an Eclipse. If one of you is very rich and wants to donate some for COVID research, please, I would also happily address this question, but maybe we'll find out in the future through some collaboration. Would be nice.
Yeah. And then, so with the mePROD approach, how do you account for protein degradation? Because this would obviously presumably change the heavy to light ratio independently of translation.
It would. I mean, for starters, I mean, we try to do it at very short time, so usually there's not much happening. But the other great advantage is because we do everything relative to this boost channel, we don't really rely on the light channel, on the light signal not changing. I mean, we call it total heavy to total because that's in the normal understanding what it is, but it will not be affected by the degradation because of this relative quantification to the boost.
Cool. And then more biological question: why did you choose the COCO2 cells, and is there any alternative?
The very simple answer is because it worked. And I mean, the first paper we sent out end of February, and I think we see how long it took in a lot of places to set up any kind of infection system. And I think this is really largely driven by this, our collaboration partner, Jindrich, who has worked in SARS in the very early days in 2003 when it just came out. And it just turned out that COCO2 are really great for these coronaviruses and they work well. We've tested out some other cell lines, and we would really love to also, of course, get into primary models, but it's very difficult in primary cells to find conditions to infect them, especially very efficiently. And of course, with all those, we're always very limited, even though we are in hospital, right? We cannot do these big experiments where you can do tox curves and then do IC50 values in 5 and more replicates. it's tricky, but I mean, we're trying, and we have quite done a lot of initiatives in that direction. But I think the papers that I see coming out very often still rely on these cancer cell lines.
Mm-hmm. And when you were looking at the viral proteins in your phosphoproteomic data, did you factor in any glycosylations as PTMs when searching that data?
We did not.
No.
Yeah, I know, and we're involved in some collaborations where people are trying to look into that. But I think with the tools, the way we run it, I don't think it would be, we would get very deep or very meaningful data.
And do you think this particular study will be useful for also finding druggable target sites on the SARS-CoV-2 for drug molecule binding, or are you really focusing on the host cell proteins?
I think host cell is much easier at this point. Of course, there are a lot of efforts going on the viral proteins, but I think what we need there is structures, right? Get ideas where we need to inhibit them and then to improve these compounds. But I think because there's so few proteins, there's no need to first identify them with our proteomics. I mean, the only thing you could imagine is, of course, if you always require, or we always see certain phosphorylations on those proteins, and these come along with a structural change, then this is something that could be or should be taken into account when kind of developing drugs for this particular conformation. But I think otherwise, the host cell pathways are a better translation target for us now. Yeah.
And have you considered applying the TMT-Pro16 channels to your samples so that you can get a bit higher throughput?
Yes, we did, and it works. That's the good part. Right. We've also testing it for where we need throughput, but again, very often we're not so much limited by the many samples, but rather the opposite, but having little material and so on, especially in the particular biological context we care about now.
Mm-hmm. And one last question. So have the enzymes associated with the regulation of glycolysis been studied using any kind of network analysis?
Yeah, I mean, how we found them, I mean, essentially all we did was to a large extent network analysis, because we thought very often, as depending on the dynamic range you have in a data set, it's often much better, instead of identifying one protein that probably changes twofold with a decent p-value, to instead identify 10, 20 proteins all in the same pathway that show a similar pattern, even if their fold change might not be as big. So that's how we actually discovered these pathways. We looked for everything that went up, did geoterms, reactomes, and all kinds of approaches. And yeah, this is how we got there. But I think this is exactly the way forward also in the future.
Okay, and one last question that's just come in. Can you provide a link for the database of the FDA approved drugs that you mentioned in your talk?
Yeah, I mean, yeah.
Yeah, great. That one came in.
Downloadable data sets. I mean, it's nothing that we specifically have there online available. It's not proprietary or anything. It's just something you can download with a lot of these network tools. Yeah.
Great. So I think that's it for the questions that came in. So thank you very much again for the excellent talk and for answering all those questions. And thanks everyone for listening. As